everyone, I'm Dr. Jaspreet and I will be sharing certain MCQs regarding the FMGE exam. The topic for today is concepts in biochemistry. Now, the first MCQ which I am taking is on mitochondria. Now, as we know that mitochondria is the it is the powerhouse of the cell. The main function of mitochondria is the ATP synthesis. Now let us see that mitochondria, it has the outer membrane which is involved in lipid metabolism. The inner mitochondrial membrane, it is involved in electron transport chain. Here the ATP synthesis takes place. Now the space between the outer and the inner, it is involved in nucleotide metabolism. The matrix, this is the matrix. Here we have beta oxidation. We have the TCA cycle. It is the tricarboxylic acid cycle. And we have the enzyme glutamate dehydrogenase, which is the enzyme involved in oxidative deamination. And we have apoptosis taking place in the matrix. So mitochondria is basically involved in catabolism, catabolic reactions. The majorly cytoplasm is involved in anabolic reactions. Now coming to the question, mitochondria they are involved in all the following except A is ATP production. Now this is the function of mitochondria, as we see in electron transport chain. There is synthesis of ATP. Apoptosis, this is also taking place in the matrix. The TCA cycle, this is also taking place in matrix. Cholesterol synthesis is the answer, as this is the anabolic reaction which takes place in the cytoplasm. Next question, it is on diabetes. Now, as we know, diabetes, we have type 1 and type 2. The type 1 diabetes, it is there is deficiency of insulin. Insulin is an anabolic hormone, and we see that the body turns into a catabolic machine. Why? Because we have deficiency of insulin. So there are all catabolic reactions taking place in the body. Now let us enlarge this picture. Shall I enlarge it further? Now, let me put it on the left side so that we can see it. So, here. Now, what happens? We see here that the adipose tissue. Here, what happens? We have all the triacylglycerol, which is metabolized, and we release fatty acids. These fatty acids, they reach the liver. Here, they either form VLDL or they are the acetyl-CoA, which is being released, is involved in ketone body synthesis. Then the oxaloacetate formed here will be used for gluconeogenesis, forming glucose. Now, the amino acids, there is a breakdown. It's a catabolic state of the body, so it is again being converted to glucose. So we have gluconeogenesis, we have ketone body formation, we have more of lipolysis going on, and these cells, they are not able to take up glucose inside. Why? Because GLUT4 receptors, which are sensitive to insulin, they stop responding. Hence, coming back to the question, let me minimize it a bit, the original size. Now, the question here, in type 1 diabetes, which of the following is true? A. Increased lipolysis. Yes, of course. There is increase in lipolysis. We have more of breakdown of the triacylglycerols. Decreased hepatic glucose output. No, gluconeogenesis increases. So, decreased protein catabolism. No, there is a catabolic state. So, proteins are being catabolized. Increased glucose output. No, glucose is not being able to enter the cell. It is a state of iceberg mechanism going on. The glucose outside the cell in excess rather than inside the cell. As the GLUT4 receptors, they become resistant. They do not respond to insulin. There is deficiency of insulin. Now coming to the third question. This is on myocardium. As we know, myocardium, it is a involuntary muscle. It is the muscle present in the heart. It is surrounded by the pericardium and the endocardium. This is the endocardium, this is the pericardium, and this is the mitochondria. Here, sorry, this is the myocardium. Now, myocardium, as it is functioning 24 hours, so it needs energy much more than what the normal ordinary cells require. So fatty acids are the ones which are being utilized for as a fuel for the source of energy. Normally in neonates and in MI we use glucose because less of the oxygen is being utilized for its breakdown. Whereas in starvation, it is the ketone bodies. So in starvation, what we have is ketone bodies. So usually it is using fatty acids. In starvation, it uses ketone bodies. Whereas in neonates, it uses glucose, and MI, it is using glucose. Now, coming to the next question. This is on glycemic index. What is glycemic index? Glycemic index is a number given from 0 to 100. Glucose given the maximum, that is 100. Why it is given 100? Because it gives maximum rise in the glucose level in our blood. We see a sudden peak and then rapidly it falls down to normal as seen in this diagram. This is for the high GI substance, and this curve, as we see, this is flattened and goes for a longer way. This is the low GI substance. So The lower the GI, slower is the absorption. They don't peak the glucose level, and for a longer state we don't feel hungry as the glucose level is maintained in our body. So coming to the choices, low glycemic index food is A. Increase plasma glucose. No. The high GI index food will increase the plasma glucose level. Yes, it has a slower absorption, or it has slower absorption. This is right. Then increase glycogen deposit. This is wrong. And it is easily digestible. Again, this is a wrong choice because it takes much more longer time to digest the products with low GI index. That's how the blood glucose level doesn't rise immediately. So coming to the last question, that is on glyceraldehyde. As we know, the glyceraldehyde, it is a 3 carbon aldehyde. It is a aldose sugar with 3 carbon. Now, we know that Benedict's test, it is the test for reducing sugars. And monosaccharides are reducing sugars. And this is the basic, the triose glyceraldehyde, which is a reducing sugar. So it will give this test positive. Now, Molish test. This is a test given by all the monosaccharides with minimum of 5 carbons. Now, glyceraldehyde has 3 carbons. So, this will be negative for Molish test. Selivanov's test. This is a test for keto sugars. Glyceraldehyde being a aldose sugar, this test will be again negative for glyceraldehyde. Gerhardt's test, this is for ketone bodies. The other test is Rutra's nitroside test. So, the answer to this question is Benedict's test. Thank you.